Hi everyone, and welcome to our video on DNA profiling. This is part of our Module 6 Part 3 series on manipulating genomes, and what we're going to be doing in this video is looking at specification reference 6.1.3c, the principles of DNA profiling and its uses. Let's go back a little bit in time first of all to the actual development of this process of our DNA fingerprinting. So in 1978, there was a scientist in Britain called Alex Jeffreys, and he was working on locating tandem repeat sequences of DNA. Now, these tandem repeats are little 10 to 100 base pair long repetitive DNA segments, but they don't code for proteins. So what they have is this core sequence, which I've shown you there. You don't have to memorize it, don't panic. And you'll notice that even though a lot of these are the same, we then have an X in there. And that can be any of the 4 nucleotide bases. So these little tandem repeats occur over 1,000 locations in the genome, and at each point they could be repeated any random number of times. So what we've got there then are these short little sequences that don't code for a protein, that have a very identifiable core sequence, that are located in a number of places through the genome, and they're just going to be repeated different numbers of times in different individuals. And some of these repeats are what we describe as highly variable. So these are the ones that we give the little acronym VNTR, variable number tandem repeats. So they're the ones that show a lot more variability than some of the others that are a little bit more stable. So what do? We want to carry out a DNA profile on someone. Well, first thing, we get the DNA. Now, the DNA is typically these days going to come from things like saliva tests, and if you've done certain little DNA tests yourself, then that's where it's probably come from. What we then do is we digest that DNA with restriction enzymes. So the restriction enzymes are going to cut the DNA at specific sequences, and then we're going to use the process of gel electrophoresis, to separate the fragments, and it's going to separate it based on the size of the fragment. We then stain that gel, and we can then see the pattern of bands that forms. And all we then need to do to identify if something comes from one person or another is just compare those banding patterns. Now, as we mentioned, we're going to use these restriction enzymes to cut the DNA sample. And the reason that we use restriction enzyme is that they're going to have these recognition sites, specific base sequences that they will cut the DNA at. Now, what we find is that those restriction enzymes cutting at those same recognition sites means that we're obviously going to be cutting at that same point in all people's DNA. But we're going to have different length fragments generated when we use the same restriction enzymes. Because if we're thinking about that idea of these tandem repeats, and we've got obviously one person who's got only 5 repeats and another one that's got 10, then clearly that restriction enzyme cutting at those same points is going to generate a longer sequence for one than the other. So what we end up with is this idea of restriction fragment length polymorphism. A lovely mouthful. So restriction fragment, i.e. the fragments of DNA made through the cutting of restriction enzymes. The length is obviously how long they are. Polymorphism, so poly means many, and morphism is like forms or types. So what we can see here is that we are generating many different lengths of DNA through the use of a restriction enzyme to cut them. Now, what that means as far as a DNA profile goes. If we've managed to collect some evidence at a crime scene, and we've then used our restriction enzymes to cut it, put it through the gel electrophoresis, and we've emerged with this banding pattern on the left there. We then have 3 suspects. We're going to take a sample of their DNA, typically through swabs of their mouth, etc. And then we're going to run those with the same restriction enzymes through electrophoresis, and then we're going to generate the individual banding patterns for our 3 suspects. How do we identify which one was the one that left their DNA at the crime scene? Match the bands. It's literally spot the difference. So all we've got to do is have a little look along, and we're just looking for the one where all of these bands match up. And if you look at suspect 2 and the crime scene, you can see that we've got a perfect alignment with all of those bands. So suspect 2 left their DNA at the crime scene. If we think about the current technique for our DNA profiling, then what we use are these things called short tandem repeat sequences, STRs. Now, STRs are highly variable short repeating lengths of DNA, and the key thing about these is that the number of them varies person to person. So not only do we have this polymorphic nature of each STR as an individual, but the number of those STRs is going to change from one person to the next. When we think about how many of these we will use in a DNA profile, then it's 13 that we currently use. So we will analyse 13 of the STRs, and when we do this, the reason we do this is that the chance of 2 people sharing the STRs at all of those 13 loci we're examining is 1 times 10 to the power 18. And if we put that into context, the world population is approximately 7.8 times 10 to the power 9. So what we can be pretty certain of in that scenario is that whoever's DNA matches, it is that person. The likelihood of it being another random person on planet Earth sharing that exact same set of STRs is so improbable. We can't say it's impossible, but it is immensely improbable. One thing we do need to be incredibly careful of when we're doing any of this DNA profiling is the risk of contamination. Because if we get even the smallest amount of DNA that shouldn't be there in our sample, we get an inaccurate profile. So this could be someone wandering through the crime scene. It could be someone in the lab not adhering to, obviously, best practice lab policy. It could be anywhere along that line. If people aren't following the right protocols, we could get contamination, and therefore the DNA profile is, to all intents and purposes, useless, because we're not going to get a pattern that matches. So how do we use these DNA profiles then? We've been talking about one quite a lot in forensic science there. So forensic science, the way you've probably heard about it is obviously the lovely CSI shows, etc. They have you believe that we can run a DNA profile in about 5 seconds flat and therefore convict a criminal by the end of the episode. Obviously, real life isn't quite like that, but we do use DNA profiles to actually convict criminals. We can then put certain people in a place. Obviously, if we're talking about sexual assaults, etc., then yes, we can get that evidence from there. On the flip side, though, we can also use DNA evidence to overturn wrongful convictions. I would love to say the criminal justice system is perfect, but it is not. And particularly in some places in the world, America, I'm looking at you, then there has been a lot of issues in the past, particularly around certain ethnicities of folks getting wrongful convictions more than others, let's say. Therefore, what can we do these days? We've got our DNA profile technology. If there's evidence still in storage, and there should be, then we can run the profile from the evidence versus our person who's currently sitting in prison, and we can overturn those wrongful convictions. And this has actually been something that's been done a disturbingly large number of times. So it does go to show that it can also help to free people who were wrongfully convicted. Something that's not particularly pleasant, but we do sometimes have to use DNA to identify victims of certain incidents. Obviously, usually we'll be able to identify by facial recognition, but in situations where that's not possible, then we will have to use DNA from whatever remains may be available. And we can also use it in things like identifying fugitives as well. People go to quite extreme lengths to hide who they are when they know they've done some rather horrific things. And one of the best examples of these are some Nazi war criminals. Obviously, post-World War II, a number of people are involved in all the atrocities in the concentration camps, made a dash for it and then have been living life in hiding. If we think we've identified one of them living somewhere in the world, we can use DNA evidence to then prove that they are actually that Nazi officer. Then we come on to the maternity and paternity tests. If you remember the days of things like Jeremy Kyle, then you know the good old paternity test. Obviously, maternity not so much, but paternity tests were the ones that were almost big business on those talk shows. Now, the reason that we can identify who the parents are is because half of a child's STRs will come from each parent. So 50% from the mother, 50% from the father. If we take that DNA from the child and then we take DNA samples from any of those potential parents, we can then just compare those profiles and we'll be able to identify which parents actually belong to that child. Now, obviously paternity tests are the ones that people tend to go for because most people don't think that you wouldn't know who your mother was. But there are some rather horrific cases where babies have been stolen from hospitals and things like this, or mixed up in those truly traumatic cases. And what we then find is we can prove who the mother is as well. Clearly, if you've given birth to a child, there's a pretty good guess that it's yours. And then we've also got a disease side. Now, in terms of diseases, we can use something called protein electrophoresis, which is all about detecting specific proteins which are markers of a disease. And a good example here is Huntington's disease, caused by the Huntingtin protein. So what we can do is detect that protein, and then we'd be able to identify someone as having Huntington's disease. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel where you can see when I next post the next video, and head on over to the website where you can find a range of other resources to help you in your A-level biology studies.